Hello everyone and welcome to Chapter 18. In this chapter we will talk about the electron transport chain as well as oxidative phosphorylation. In this first video we will discuss the electron transport chain. So first we'll discuss the overall reaction that contributes to the full electron transport chain, and then we'll talk about the individual complexes. In the next video, we will discuss the sort of next step of this process, which is oxidative phosphorylation, where this proton gradient that is generated through the electron transport chain is ultimately used to help finally make ATP to be used as an energy currency by the cell. But first, we had to discuss the electron transport chain. So where does the electron chain transport chain fit in our overall picture where we're looking at glucose catabolism? So we started off with glycolysis, where we broke down a glucose molecule into pyruvate, some ATP, and some NADH. We then took that pyruvate, put it through the pyruvate dehydrogenase complex to generate some carbon dioxide, an acetyl-CoA, and another reduced electron carrier, or NADH. And finally, we discussed the citric acid cycle, where we took that acetyl-CoA, we generated some more CO2, and some more energy in the form of GTP, as well as more reduced electron carriers, NADH and FADH2. So the ATP and the GTP that were generated in these catabolic processes can be used directly as an energy currency, but the cell has to now do something with all of those reduced electron carriers that were formed in these three processes, and that's where the electron transport chain comes in. The electron transport chain is a method for the cell to use to generate a proton gradient using all of those reduced electron carriers, in particular NADH and FADH2. After this proton gradient is generated, then oxidative phosphorylation comes in, which uses that proton gradient to finally synthesize ATP. I also want to make a note that these reactions that are written here are not balanced reactions. They're just here to give you a sense of a summary of sort of what sort of sources of energy we've generated so far during all of these different catabolic processes. In terms of this picture, we have our reduced electron carriers here that are coming in from glycolysis and the citric acid cycle, as well as the pyruvate dehydrogenase complex. And during the electron transport chain, we do use some oxygen to generate water, and then finally during oxidative phosphorylation we use the proton gradient that we generated to make that ATP. So where is this all occurring? So just as a review, we already know that glycolysis took place in the cytosol, and then the pyruvate dehydrogenase complex and the citric acid cycle, all of those enzymes were located in the mitochondrial matrix. So where is the mitochondrial matrix? As you can see from this diagram on the right showing a mitochondrion, the matrix is the lumen in the center of the mitochondrion. So a mitochondria actually has two different membranes. There's an outer membrane and an inner membrane. So the matrix is inside of the inner membrane. In between the inner membrane and the outer membrane, there's a space referred to as the intermembrane space. So the like I just mentioned, the PDC and the CAC, the pyruvate dehydrogenase complex and the citric acid cycle, are taking place in the mitochondrial matrix. And then as we move on to the electron transport chain, all of the different complexes involved in the electron transport chain are located on the inner mitochondrial membrane, so right on that, essentially the wall that surrounds the matrix. And oxidative phosphorylation is also going to take place there at the inner mitochondrial membrane. So let's first look at a larger overview of everything that's going on in the electron transport chain. So the first step is where our reduced electron carriers are going to enter, either through complex 1, which is where NADH comes in, or through complex 2, which is where FADH2 comes in. Next, all of the electrons are then transported by coenzyme Q, which is also written as CoQ. Sometimes it's also just written as Q. It's also called ubiquinone. And these electrons are eventually all brought to transport 3. At this point, the electrons are then moved over to cytochrome C, where they're brought over to complex 4. And during a lot of these different steps, protons are pumped out of the matrix through that inner mitochondrial membrane to the intermembrane space to create a proton gradient. So all of that is also summarized in this figure. And so I think this figure is a really helpful place to start, and it contains most of the information that you will be expected to know in terms of what's happening during the electron transport chain. So complex 1 is depicted here in this sort of gold color. This is where the reduced NADH comes in and drops off its electrons. NADH itself gets oxidized back to NAD+. Through this process, 4 protons are pumped out into the intermembrane space from complex 1. Complex 2 is shown here in blue. Complex 2 is where FADH2 enters the electron transport chain. And as you recall, the FADH2 that was generated during the citric acid cycle was part of that succinate dehydrogenase enzyme. And so during the course of the oxidation of succinate, that's when we generated FADH2, and those electrons are then transferred over to complex 2 to enter the electron transport chain, and FADH2 can be oxidized back to FAD. Those electrons, like we mentioned, are then transported by coenzyme Q, which is shown here in this as this little Q, where they're brought over to complex 3. And then in complex 3, these electrons are transferred over to cytochrome C, which is shown here in green. And during this process, 4 more protons are also pumped into the intermembrane space from the matrix. Finally, cytochrome C brings the electrons over to complex 4, where the protons are used to help generate a water molecule, and this is the point finally in the electron transport chain where oxygen is required. And during this process, 2 protons are further pumped out into the intermembrane space. And so keep some of these sort of numbers in mind, and we'll do some math at the end to sort of add up all of our protons and where they come in. So let's start by, and I just again want to say that as we go over each of these complexes, I'm going to include more detail that will hopefully help you understand how these complexes work and why they do what they're doing. But the level of detail that you're expected to know for the quizzes and exams is less in general than the full details that are shown on these slides, as you'll see as we go through the practice questions later during class and on your own. So that being said, let's start by talking about complex 1. During complex 1, like I mentioned, this is where NADH is going to enter in, and so NADH itself is going to get oxidized to NAD+, and it is transferring its electrons over to coenzyme Q. So if NADH is being oxidized, coenzyme Q, as it accepts those electrons, is going to get reduced into CoQ-H2. And like I said before, coenzyme Q is also sometimes just written as Q in the oxidized form, and in the reduced form, it's sometimes written as QH2. And so ultimately, as these 2 electrons are passed from NADH to coenzyme Q, 4 protons are also then pumped from the matrix into the intermembrane space. And this is the place where NADH is entering the electron transport chain. Complex 2, like we mentioned, is where FADH2 is going to enter the electron transport chain. And complex 2 contains that succinate dehydrogenase from the citric acid cycle that has that bound FADH2 in it. So the FADH2 is again going to be passing off its electrons to an oxidized coenzyme Q to help regenerate, oxidized FAD and to generate a reduced coenzyme Q. This process of transferring the electrons from FADH2 to coenzyme Q doesn't generate enough of a free energy change to actually pump any new protons from the matrix to the intermembrane space. But it is the entry point it's the only entry point for those FADH2 electrons. And later on, in complexes 3 and 4, these electrons can help to generate that free energy change to pump the protons into the intermembrane space. It's just not happening in complex 2. And that's different from in complex 1 when NADH was entering and there were protons that did get pumped into the intermembrane space. So let's now take a minute and talk about coenzyme Q. So coenzyme Q is a small organic molecule. It's not protein. It contains a 6-membered ring that has these 2 ketone groups here and these 2 double bonds here in the oxidized form. Again, this molecule is also sometimes called ubiquinone or just Q. It also can be in the fully reduced form, where we have hydroxyl groups here instead of the ketone groups, and we have 3 double bonds in our 6-membered ring. And then that's the fully reduced form, so we have QH2 or coenzyme QH2. And there's also a semiquinone form, or this radical form, when there's only one electron that's been donated. So the fully reduced coenzyme Q has two. Partially, this sort of semiquinone form has just one extra electron, and the oxidized form has the fewest. So the coenzyme Q itself, it is able to diffuse, it exists within the inner mitochondrial membrane, and it can diffuse laterally within the membrane. And so what it does is it carries 2 electrons and 2 protons from complex 1 and complex 2 over to complex 3. And just to review, coenzyme Q, or Q itself, is the oxidized form, and coenzyme QH2, or QH2, is how it's written in the reduced form. And then you can see over here how it is written in the radical or semiquinone form. All right, so that brings us to complex 3. Complex 3, like we mentioned, is where coenzyme Q is carrying over those electrons from complexes 1 and 2. And so at this point, coenzyme Q is in the reduced form, CoQH2, and it's going to help to transfer over those electrons over to cytochrome C. And the oxidation state of cytochrome C is represented by an iron, which is contained within cytochrome C. So when it's iron 3+, it's the oxidized form. And then we also, when it accepts those electrons from coenzyme Q, it's in the reduced form. But it's important to note here, we need 2 cytochrome Cs for every 1 coenzyme Q, because coenzyme Q is able to carry 2 electrons, but each cytochrome C is only able to carry just 1 electron. And so during this process of transferring the electrons is actually relatively tricky, partially because coenzyme Q is a 2 electron carrier, but cytochrome C is only a 1 electron carrier. And so the process of transferring the electrons from one to the other is known as the Q cycle, and that's what's occurring in complex 3. So during this process, for every 1 coenzyme Q that gets oxidized, 2 cytochrome Cs are getting reduced. And also during this process, 2 protons are getting removed from the mitochondrial matrix, and 4 protons are getting released into the intermembrane space. So let's take a closer look at how exactly that Q cycle works. So first we start here in cycle 1. So a reduced coenzyme Q enters from either complex 1 or complex 2, and it will bring its electrons into complex 3. So what we're looking at here is complex 3, where we have cycle 1 and cycle 2 occurring. So once the reduced coenzyme Q enters into complex 3, it releases these 2 extra hydrogen molecules. So the 2 protons get released into the intermembrane space, and then there are 2 electrons that have to get removed as well. So one of the electrons can get sent right over to a cytochrome C, and at that point the other electron is actually handed off to another coenzyme Q that starts off in the oxidized form and ends up in the semiquinone form, because it's only accepting 1 electron from our original coenzyme Q, which is shown over here. So that's part of cycle 1. In cycle 2, we have yet another reduced coenzyme Q entering in to complex 3 from complex 1 or 2. So the beginning of cycle 2 looks the same, where our reduced coenzyme Q that entered in from the outside releases 2 protons into the intermembrane space and hands off 1 more electron to cytochrome C. But at this point, the other electron is now handed off. So recall that we made the semiquinone Q during cycle 1. That second electron from our newly entered QH2 can now be handed off to that semiquinone coenzyme Q to fully reduce it. And at that point, once it's fully reduced with those 2 extra electrons, now 2 protons are pulled in from the matrix to fully reduce this coenzyme Q into the fully reduced coenzyme QH2, and that completes cycle 2. So overall, you can see here how 2 protons are getting released into the intermembrane. I don't know if I said 2 or 4. 4 protons in total are getting sent out into the intermembrane space, and 2 protons are being pulled in from the matrix. And so that's the Q cycle. So at that point, the electrons have been passed over to cytochrome C. And so let's see how that, or let's talk about now what cytochrome C is doing and how it works. So cytochromes in general are proteins, and they have heme iron groups, which contain iron 2 and 3. And these heme groups are able to absorb visible light. So that's part of, so chrome is in their name, which refers to color. And so that's part of why they're named how they are. And so you can see in this diagram here, the protein portion of cytochrome C is shown as this green color, and the heme group is shown here in red, and it's in a planar form, which is why it's shown as this sort of red line. So cytochrome C itself is a soluble protein, and it's a peripheral membrane protein, so it's able to associate with on the intermembrane space side of the inner mitochondrial membrane. It associates with the membrane, but it's peripheral, so it's able to move away from the membrane into the intermembrane space itself, as well as to interact with the the intermembrane space outer portion of that membrane. And like we just saw, each one cytochrome C molecule is able to accept one electron in complex 3 from that coenzyme Q. And each cytochrome 3 then delivers that electron over to complex 4, which brings us to complex 4. So in complex 4, we have 2 cytochrome Cs that enter in the reduced form, and they are going to get oxidized back into the iron 3 form. And during that process, 2 protons are removed from the matrix and combined with an oxygen molecule to make water. And so, like we said earlier, this complex is the reason that oxygen is required for this oxidative phosphorylation process. Without oxygen, complex 4 wouldn't work, and the electron transport chain would not be able to be completed. So like we mentioned, this process in complex 4 also is going to, remove 2 protons from the matrix, which help to form that water molecule. And it also is going to pump 2 protons from the matrix out into the intermembrane space to help contribute more to that proton gradient. I also want to mention here, double stoichiometry is actually required because here when we see just one water molecule being formed, it requires a half of an oxygen molecule, which doesn't actually exist in real life. So in real life, you have to double everything up. So you would have 4 cytochrome Cs that are reduced and 4 protons with 1 oxygen molecule help to get, so then we have the reoxidized 4 cytochrome Cs to make 2 water molecules. And that way we have whole numbers of all of our molecules. So overall, what did we just see? So for every 1 NADH that gets reoxidized back to NAD+, that's going to move 10 total protons from the matrix to the intermembrane space. For every 1 FADH2 that gets oxidized back to FAD, that's going to move only 6 protons from the matrix to the intermembrane space. And part of the reason we see this difference is, if you recall, FADH2 enters through complex 2, and in complex 2 we did not contribute anything to the proton gradient, versus NADH, which enters through complex 1, where we do see a contribution of protons to that proton gradient. And finally, now that this proton gradient has been generated, it is able to be used to help synthesize ATP during our oxidative phosphorylation, which we'll discuss in the next part of this chapter. But at this point, now you should be ready to answer the electron transport chain practice questions.